and this will be heavily, I guess, drug development based from the technology side of things. So, okay, I work for Allison, that's our disclosure slide. So today I'll walk you through a bit of an overview of AAV gene therapy, discuss some of the pivotal clinical trials and look at some of the challenges and what we're doing to try and overcome these. So when we think about gene therapy, we need to recognise that it has quite unique biological characteristics compared to other protein-based therapies like antibodies. But ultimately, it consists of two basic elements, genes and a way to deliver them. It's really important for us to understand the basis of the disease we're treating to ensure that we are delivering the optimal therapy in the safest possible way. So if we consider delivery methods, they basically fall into two categories, viral vectors and non-viral vectors. I'm obviously here to talk about a viral vector, so Adeno-Associated Virus, but we do need to recognise that there is the growing field of non-viral delivery, particularly LNPs and mRNA technology. And what we can see now is a bit of a synergy between both these modalities, whereas previously they were moving forward in quite independent ways. So which vector delivery method you choose will be dependent on these things listed at the bottom of the slide here. So the choice will be dependent on the cell state of the cells that you're trying to target, whether they're dividing or non-dividing. The tissue, obviously there's some tissues that are much easily targeted, like the liver, compared to, say, crossing the blood-brain barrier. You also need to consider the cargo capacity. That will dramatically determine what vector you choose. You need to consider the immune response. In some cases, you might need a strong immune response, say with cancer therapeutics. Most other times you'd like to avoid that immune response, and we'll talk quite a bit about that today. And you also need to consider how you're going to deliver it, whether it will be in vivo, systemic, or directly injected into the target organ, or ex vivo. So we're here today to talk about Adeno-Associated Virus. So this was first identified in 1965 as a contaminant of an adenoviral prep. So this is the first image we have of adenovirus, of AAV, sorry. And so you can see here this big virion is the adenovirus, and all these small ones are the AAVs. So this is why it's called Adeno-Associated Virus. It's a single-stranded DNA virus that does require the help of another virus, such as adenovirus or herpes simplex virus, to complete its life cycle. In the field, it's generally viewed as non-pathogenic, and this comes from the notion that as of yet there's no known disease that's been identified. However, up to 80% of the population has neutralizing antibodies to at least one of the serotypes of AAV. It's viewed as a less potent initiator of the immune response compared to some vectors like adenovirus, but we can't discount the fact that we do mount an immune response, and this becomes apparent in the clinical trials we'll discuss. The majority of the vector genomes persist as episomes, so extrachromosomal DNA, rather than integrating, but it needs to be noted that AAV does integrate, and early papers show that AAV, wild-type AAV, can integrate in a site-specific manner, but not site-specifically all of the time, and recombinant AAV it does integrate, but not in a site-specific manner. The neat thing about AAV is that it can be pseudotyped or coded with different capsids, and this has opened up a world of possibility for us in the fact that we can target many different tissues. So we need to dive a little bit deeper into the components of AAV, and basically there are two that we need to be aware of. There's the capsid. So it's the capsid that defines tropism, so what tissue you're able to target, and also the transduction efficiency, so how effectively can you deliver the DNA payload into the nucleus. So it heavily determines the path that the virus takes through the cell. It's also the part of the virus that we can readily detect an immune response to, and this is through either neutralizing, binding or neutralizing antibody assays, or looking for a T cell response. I just wanted to point out that when AAV is assembled, the capsid assembles first, and then the vector genome is inserted through this five-fold wall. And we like to think that this is a really efficient process, but we're coming to recognise more frequently that it's not just the viral genomes that can be packaged, and again we'll talk further on that. So the second component of AAV is the viral genome. We need to note that its size is about 4.7 kilobases. So this is the packaging capacity of AAV, and that becomes important when you're considering different genetic diseases that you might want to target, and the size of the gene that you need to be replacing, because you need to be able to fit it if you want to use AAV into this size, or you need to get creative about how you're going to deliver that therapeutic. So ultimately it's a fairly simple genome that utilises a unique set of promoters and splicing sites and non-consensus coding start sites to produce the 4 rep proteins that are required, and the capsid proteins for the assembly of the capsid. I'll just highlight a couple of key things here. So what flanks this genome are these ITRs, or inverted terminal repeats. So these are DNA hairpins, and they are the origin of replication and the primary packaging signal. The rep proteins are unsurprisingly important for DNA replication and packaging, and then obviously the cap protein produces the 3 components of the capsid, the 2 minor coat proteins, VP1 and VP2, and the major coat protein, VP3. So I can talk till the cows come home about this if you want after the presentation, but those are the key elements that you need to know about. So the other important thing to pay attention to is that there is obviously a difference between wild-type AAV and recombinant AAV, and this is effectively the evolution of the field. So I just talked to you about the genome of wild-type AAV, and the early work in the 80s and 90s was focused on how to shift this into a recombinant format. And one of the key things that the researchers back then, including Jude Samolsky and Nick Waziska and all of those leaders of the field, was that the only element of the wild-type genome that is required in the final AAV product is the ITRs. So these flank your transgene of interest, and you include in there regulated elements for regulation of that gene expression to produce your protein of interest. So we now understand what recombinant AAV is, but we also need to take a look at how we do make AAV. So in industry, we have lots of fancy terms for this, and basically up the top here is the process that CMC development takes. So we start with certain starting materials and proceed through these steps until we have our final clinical product. So the first important thing to think about is how we actually make this AAV. So the original work, and what is still currently quite a workhorse for AAV manufacturing, is a 3 plasmid system. And this is where we have our vector genome, so again, it's the transgene flanked by the ITRs. A second plasmid that will express the rep and the cap genes. And I just want to note that the majority of the clinical products contain the ITRs of AAV2 and the rep of AAV2, primarily because that was the first AAV that was really well characterized. And this second plasmid is where we can switch out the capsid and basically the capsid gene basically change the code of the protein. The third plasmid contains the adenoviral help. So in the early days, AAV was made with wild-type adenovirus as the helper function, but it was realized that there are just certain elements of adenovirus that are required to be able to produce recombinant AAV. So this part of the manufacturing process is a place where there is a lot of innovation that occurs. One important innovation over the last number of years came from Torchlight Therapeutics, where they developed a novel synthetic DNA manufacturing system that does away with a standard bacterial plasmid format. And why that's important is that here we obviously need to recognise that these plasmids contain bacterial genes in the backbone there, and we don't want to include any of those in our final product. So they developed this system which does away with that. They refer to it as the doggy bone DNA system, and it really is sort of a leap forward in our ability to produce AAV. The other place that people are focused on is trying to use different combinations of these plasmids, so you can split the rep and cap to try and reduce your chance of producing replication competent AAV. So ultimately, all the work at the front end is trying to reduce your immunology and genotoxicity risk, and trying to reduce partial genomes that could be packaged. So the next part of the process incorporates the upstream and the downstream processing. So what we do is we take our plasmids or our doggy bone DNA, we transfect that into cells. A lot of the early work was done with adherent cell cultures, so like HEC293 cells. Obviously this rapidly becomes quite a manual challenge when you need to produce clinical grade vector. And so the field has put a significant amount of effort in trying to develop suspension culture systems to improve this ability to scale to clinical grade. And currently there are two general systems, a mammalian culture system and an insect cell lines, or a baculovirus production system, but I won't go into those details. So a lot of effort at this front end involves trying to process adjust to reduce any incorporation of DNA that exists in these cells in your final product, trying to prevent that encapsulation and trying to maximize the number of particles that will contain your full genome. After your upstream process, you need to be able to purify your vector. In the early days, it was done with ultracentrifugation, but now we frequently use, particularly at scale, chromatography, and this allows us to maximize yield, but also a significant amount of effort in the field has gone into trying to remove those empty capsids and the partially filled capsids, because as I said, we'd like to think this process of production is very streamlined, but we are beginning to recognise that we're packing in more than we bargained for. The final part of the process is the release testing, and this is where we need to establish an accurate titer, so how many vector genomes are there per mil, and we do this by qPCR or droplet digital PCR. We need to get an accurate read on the particle titer, and so we can do this by ELISA and a number of other novel methods, and this allows us to assess that empty to full ratio more accurately. And why this is important is obviously you don't want to be putting in an empty virion, you're just adding an antigen load that you don't necessarily need. The release testing also measures other contaminants such as any contaminating plasmid DNA, any of the host cell DNA, whether there's replication competent AAV, whether the drug product is aggregating and how stable it is. And a key component of this is also to test potency, because we do need to understand the batch-to-batch variation. Okay, so I know this slide is really busy, and I'll just refer you to this great paper that came out fairly recently. And so what the one big shift in the field is using new technology around NGS, both short and long read sequencing, to be able to actually quantify and qualify what is being packaged. And so all these images are showing is taking your vector product, doing next generation sequencing on it, and seeing where those reads align along your vector genome. And so I know it's tiny, but there's an ITR here and an ITR here, and this is the backbone. And you can see the majority that is packaged is the transgene between ITRs, but there is still contaminating DNA that does get packaged. And also, it's not just full-length genome that is packaged, but as partial genomes as well. And it's really a step ahead for the field, where we can now characterize the different components and use this information to try and modify the arrangements of the plasmids that are going in at the front end, but also establish good quality data batch to batch, which will become important when you see variation in trial results. So that's AAV and how we make AAV. The other important thing to understand is how you get from outside the cell into the nucleus and your protein that is expressed, because this is an area of fairly intense research. So this is the image of a cell, and whilst it could be viewed as a fairly straightforward path, a lot of these components are still a bit of a black box for us, but that also presents opportunity to try and improve this process. So the first challenge that AAV experiences is the potential for neutralizing antibodies to block entry. We then need to get the target the capsid to the right cell type and allow it to bind. It binds through receptors, co-receptors, and binding attachment factors. It enters the cell through endocytosis, and whilst this image is just showing one pathway, it can take multiple pathways. It needs to then traffic through the endosomal compartments. This is essential for functional transduction, because if it doesn't take this path, you won't get gene expression. It's in these endosomal compartments that the capsid changes formation and really the internal parts that contain the nuclear localization signals directed to the nucleus. I just wanted to point out a key feature here is that obviously along this path you have degradation and presentation on MHC, so initiating an immune response. There's also an innate immune trigger from both the capsid as well as the vector genome that we need to be aware of. So the capsid passes through the nuclear core into the nuclear pore into the nucleus, where it uncoats, and then you need to convert your single-stranded DNA into a double-stranded DNA and have transcription and your therapeutic protein expressed. So we can consider, I know this is really busy, but I'll just point out a few things. So we can consider all of these places as areas of innovation and opportunities to improve the process, because ultimately we do know we're delivering a lot of vector to cell, but we're not necessarily having all of those vectors functional, and we really need to improve that process. So we can try and avoid the neutralizing antibodies by screening, but also technology around removing the antibodies or developing capsid mutants that escape them. We can improve the binding and this pathway to the nucleus. We can also focus on the transgene and make optimizations here where we look at not just, we can look at the genome configuration, whether we want a single-stranded format or a self-complementary format, which basically allows us to have a double-stranded genome there early on. A significant amount of work is done on, is focused on optimizing promoters and the transgene, but I don't have time to go into those details. We also can look at, and what becomes evident is this, modifying the immune response through inhibitors or modifications will really further advance AAV gene therapy. So I'll just dive into a couple of these opportunities for improvement, primarily around capsid development, because that's where I've spent a lot of my scientific career. And so this is a great review from a team at ASK. And in this figure, what they're showing is a variety of ways that we can modify the capsid just to try and improve targeting and potency. I'll pick out a couple of these. The first one is capsid shuffling. And so this is where we're harnessing the power of rapid evolution to identify novel variants. So what we do is we take a number of parental AAVs, chop them up. There's a high degree of sequence similarity between serotypes, and we're relying on that for them to come back and reassemble in completely random arrangements, which we then make vector from, so we have a wild type library, which we can then use to push it through an evolution process. And here you can use multiple means of selection. Could be on your favorite cell line and your favorite animal, combinations of both, including neutralizing antibodies to try and find the variants that will ultimately escape them and be your key variants to characterize. And these figures down here are from a paper where we used this process in an in vivo model. So it was a humanized mouse model. So the liver contains human cells. And what we did was identify a variant called LKO3 that showed a significantly higher transduction in primary human hepatocytes, both in vitro and in vivo, compared to what was at the time the gold standard AAV8. So this figure here shows the mouse liver, which contains both mouse and human hepatocytes, and AAV8 is really good at getting into the mouse cells, but not so good at getting into the human hepatocytes, whereas LKO3, the converse occurs, and you can get up to, I think, an 8-fold transduction in the human cells compared to AAV8. So the other method of capsid engineering that is heavily used is peptide display. And so what we do here is we identify a region on the capsid that can accept peptide insertions, and these we can then generate a library of these. So it can be up to or more than 5e⁷ variants, which allows us to have a massive sequence landscape to hunt through for variants that have improved compared to the parental. And these can be screened through mice or NHP. And what I'm presenting here are two great papers that have done this exercise whereby looking for variants that are better at transducing muscle cells. So at this time, AAV9 is currently the favorite serotype for transducing muscle, and this group from the Broad Institute used a GFP as a reporter construct, and they've identified
a myotropic AAV, which, these are different muscle cells. So this, if we just look at the quadriceps, we can see that their new myo AAV dramatically improves transduction compared to AAV9. And here the team in Germany used a luciferase reporter, so they're looking for bioluminescence, and it's showing a similar thing where across different muscle types you can see a dramatic shift in transduction between AAV9 and their lead variant. So this is obviously an area of intense activity, and we're starting to see more of these novel variants move through the system. So now I'll take you through some of the gene therapy clinical trials, and I'm sure you're probably aware of these, but I think it's a good place to start with looking at the early trials and some of the things that were learnt from those. So this first trial is the trial that came from Dr. Catherine High's group in Philadelphia in 2006, and they were treating haemophilia B patients by delivering factor IX in an AAV2 vector. And I talk a lot about dose, so I just highlight the dose along the way. So I believe it was a 2e12 vg per kilo dose. And it was the first time, in this figure we're seeing factor IX activity and liver enzymes. So the red line is factor IX activity, and this was the first time that it was demonstrated that you could successfully transduce the liver, and they saw expression for 3 weeks before a decline. And that coincided with an increase in the ALT-AST response. And so this was indicative of liver inflammation and some degree of toxicity, although that rise in ALTs was asymptomatic. So that was the first observation that came from the trial. The other observations we can think about are that the presence of neutralizing antibodies do block transduction, and so these are 2 patients and they had different neutralizing antibody levels before gene delivery, and you can see differences in transduction. The other observation is that there was a CD8 T cell response to the capsid antigen as detected by ELISpot. So this work showed that there was subclinical liver toxicity that was observed by the decline in factor IX and increase in ALT in the presence of capsid-specific T cells, and this had been unpredicted from the preclinical models, but it's what the whole field is focused on now to a degree. So the second trial that was a real success that built on the success of the first one came from the collaboration between UCL in the UK and St Jude's Children's Hospital here, and they made a number of changes, including swapping serotypes. So they used AAV8, which had been showing great transduction in the mouse liver. It also, in general, has a lower seroprevalence in the population. They screened for pre-existing neutralizing antibodies, and that was an inclusion-exclusion criteria for the trial. They used a more potent expression cassette by using a self-complementary genome rather than the single-stranded, so that gets over that rate-limiting step that I talked about earlier. And they were prepared to look at, because of that first trial, at ALT and be reactive with steroid treatment. And this is well written up in these 2 New England Journal of Medicine papers. And what this is showing is that we've got factor IX in the orange and the ALT rise and the delivery of the steroid. And so in this first patient, there was a high rise in ALT and then delivery of the steroid. But when Emmett talks about this, they talk about, trying to decide when to deliver that, and perhaps a delay caused this drop in expression. However, in this second patient, there was a rise in ALT and then a delivery of the steroid and maintenance of the factor IX. So this is where the sort of evidence around steroids in some respect protecting the gene delivery has come from. And in the following paper from this work, they followed the patients out, and this is just showing that when you look at bleeding episodes in these haemophilia patients, that you see benefit from gene therapy. So these early trials obviously sparked significant interest, and a number of trials. The space for haemophilia gene therapy got very busy. And what I wanted to highlight here are a couple of key things. From this review, they've listed the trials in generally chronological order, and what I wanted to take note of is that frequently what occurs now is the removal of CpG motifs from the vector genomes, because we know that CpG can initiate innate response through toll-like receptor signaling. So over time, this has become a kind of a definite that you should do with your vector genome. We see a shift from the early trials where we saw transient expression to more stable expression. And the other great leap forward was the inclusion of the naturally occurring factor IX Padua mutation into the transgene, and you immediately see a jump in factor IX activity when that is included. And so uniQure's first product didn't contain that mutation, but then they switched to the Padua mutant, and this is what became the FDA-approved drug, Hemgenix. I wanted to point out that it's an AAV5 vector, and I believe the delivery is at around 2 × 10^13 VG per kilo. So, whilst I don't have time to talk about haemophilia A, it followed a similar story to haemophilia B, although it's challenged by the size of the genome. There's a product that also is an AAV5 vector, delivered at a fairly high dose, that is in the clinic. So with these trials, and this is reviewed in this this Nature review, with these trials, so they're occurring around 2007, 2011. After that, we subsequently see a dramatic increase in the number of AAV trials that are occurring. And the other shift in what we see is a shift in the novel capsids that are being used in AAV gene therapy trials, from early days where it was AAV2-like to other naturally occurring serotypes and even novel variants. Okay, so I've obviously focused heavily on systemic delivery of AAV, and we do need to mention that there are approved, FDA-approved therapies for eye disease in Luxturna, and there's also a huge number of trials that are occurring for direct delivery to the brain, but today I'm just mainly talking about the systemic delivery. So one of the areas that intense interest was in, it was to deliver for the treatment of neuromuscular disease. And I'm sure you're all aware of SMA and the SMA trials, but I'll just quickly walk you through this. So SMA is a devastating neurodegenerative disease with the progressive loss of motor neurons, so that these children develop progressive weakness and inability to sit independently, and respiratory insufficiency, which leads to death or permanent ventilation prior to 2 years. So it was amazing to see this trial emerge from Dr. Mendel's group in Ohio in 2000. It reported here in this report in 2017, where they delivered a single dose of an AAV gene therapy and saw dramatic effects. And it's an AAV9 vector, and in that first phase of the trial, I believe they were using around 2 × 10^14 VG per kilo. So these are the remarkable results. References are down here, but I just wanted to point this one out, where this is a survival curve, and the red is following the natural history of the disease, whereas the blue and the yellow lines are normal children and children's treated with the AAV gene therapy. And you know, it's a stark difference, and really incredible to see patients that would previously have passed away by the age of 2 are now able to walk. So these incredible trials are what delivered the Zolgensma, which is the first FDA-approved systemic gene therapy. And to date, there's been up to 1,400 patients administered this therapy, and it's really viewed as a cure for SMA. Because we do need to talk about challenges, we need to note that up to a third of patients that receive this dose of around 1e to the 14 have at least one adverse event of hepatotoxicity, and it is listed as a known risk on the documents with the drug. So this brings us to talk about these adverse events, and so with the increasing number of trials, naturally we are seeing an increase in the reported adverse events, and so there's a number of adverse events that are common, and this you can see include those elevated liver enzymes, and then there are SAEs that are rare, including, and I'll just highlight severe liver dysfunction, TMA and myocarditis. And this is the place where the field is discussing intensely at the moment, and it is the major challenge that we do need to overcome. This is the slide that you can find from the FDA website, where they initiated discussions to try and bring the field together and solve or understand these challenges. And they focused on the hepatotoxicity that has been seen in a number of the systemic trials with a variety of vectors, of serotypes, as well as the TMA. I won't talk about neurotoxicity in the direct injection for the brain trials, but I did just want to note what is clear is that high dose is becoming correlated with these adverse events, and it is an area that we need to focus on. So sadly, out of there have been 11 reported deaths across 8 trials. This is from this review here, and subsequent to this table, there have been 2 patient deaths after treatment with Zolgensma. It's becoming clear that that high dose leads to a higher risk of immune activation, and we do need to be aware of and consider whether their underlying liver disease is also potentially exacerbating the problem. So this immune-mediated toxicity remains to be the major challenge of AAV gene therapy. I've mentioned high dose quite a lot, and I just wanted to highlight this. So high dose obviously means a huge viral load, and from this review they considered how many cells are in the human body, and at a dose of 1e to the 14 in a 70 kilogram person, how many viral particles would that be? And apparently it's 7 quadrillion viral particles, and this turns out to be about 100 viral particles per cell, which is a fairly massive onslaught. And so it's no surprise that the immune system is responding. There's a lot of great reviews out there, and this one is particularly well written. And the author focuses on the immune response at the different stages, and the disorders that are arising from those immune triggers. So from the innate response and the complement-driven disorders around TMA to the the adaptive response with T cells, effectively killing transduced cells, so seeing the result of hepatotoxicity and neurotoxicity. So where we're sitting is that there's a fine balance between that we need to tread between dose, delivering an effective treatment, and immunogenicity. Briefly, I need to mention one of the other risks that we are aware of, and this is the oncogenic risk due to integration. There's been a lot of focus around looking at integration events in preclinical models, so in mice and in large animals. In some of the mouse models, it's complicated by the age of delivery, so whether they were treated as neonates or adults, the background of the disease phenotype that exists in those mice. But here I'm just showing the table of large animals, and wanted to highlight these two later studies in dogs where they were out to a significant number of years, and whilst they did see integration and clonal expansion in one of the dogs, but I could be corrected on that, they saw no liver nodules or oncogenesis. So it is a risk we need to be aware of, and it remains particularly important when we're delivering vector systemically and we're getting that huge onslaught into the liver. So paying attention to HCC is also in the clinical protocols. So how can we mitigate risk? We can look at it in two buckets. So I'll just briefly talk about trial design. Obviously there's inclusion and exclusion criteria that are considered, the first one being neutralizing antibody screening, and we can discuss that further if you'd like. There's a definite need to understand the natural history of the disease phenotype you're treating, particularly if there's underlying liver disease and you're delivering a huge amount of vector systemically. Also considering pre-existing or new viral infections that might exacerbate any immune reaction. There needs to be a preparation for using steroids or other immune modulators to overcome any of the challenges that arise. And people talk about making sure that dosing occurs in centres where there's experience with gene therapy to try and be ready to solve any of these presentations of problems. So the other place where we are mitigating risk is drug design, and this includes trying basically to increase potency, and we can do that by modifying the transgene or the capsid. We want to focus on improving specificity so we can avoid targeting the liver if necessary, and we can do that by, again, capsid or route of administration. We also need to really focus on manufacturing and reducing that antigen load by removing the empty capsids and reducing those impurities just to generally reduce risk. The other area that we can focus on is improving the preclinical models that we have to basically understand this immunogenicity risk, and particularly make sure that they correlate to what's going to happen in the human setting. So I'll just whiz through the next slides because I have covered this in the previous part of the talk, but capsid engineering is a place where we basically trying to expand the sequence space through which we can identify lead variants, and we can do that by the methods I've talked about previously, but also by rationally designing them. And there's the buzzword of the day is machine learning, and these all get incorporated together where we can improve targeting and detargeting, massively improve that functional transduction so that we don't need to deliver 100 viral particles to a cell, but we just need to deliver a few and know that they are going to produce the protein that you're interested in. And the other key part is to identify models that we can really predict what's going to happen in the human setting. If we think about the genome, there's a key number of places that we can modify to reduce risk, particularly thinking around the promoter, making sure that it's cell type specific, making sure that it is balanced so that we don't want a ubiquitous strong promoter all of the time, because that increases your chance of the risk of insertional mutagenesis. But we're also looking at gene regulation through promoter modification, and the team at AskBio have a system where they're heavily focused on developing regulatable promoters. The other area where we can limit reactivity, I already mentioned, was around the innate response and removing CpG, but other motifs. We can include sites such as microRNA binding sites to reduce off-target effects. We can include elements that increase the potency, so again, you're not going to need as much vector to be delivered, but also to maintain durability, because we need to recognize that this is, like, at this stage, a one-shot therapy. And again, model systems again. So ultimately, all of this is to try and lower the dose and reduce that immune reactivity risk. I won't talk about this because this is manufacturing, and I've mentioned a number of places where we can reduce risk. And I think this is my last slide. So I wasn't quite sure how to finish this slide, because I know it should always be on an upbeat note after talking about challenges. And I just leave you with this comment from Jude Samolsky, who from the Columbus Children's Foundation website. And then that's a foundation where its aim is to deliver gene therapy for very rare disease. And they're doing an amazing job at doing that. So I've been in the field for a while, doing what I can, but it's been a real privilege to see it kind of change over time to the point where we now actually have cures for these children. So I thank you very much for listening. I lost that slide there, but that was just talking about AskBio, which is based in North Carolina. And we're a gene therapy company and have a number of gene therapy trials running. So thank you for your time.
Thank you very much for this excellent presentation. We have time for a couple of questions, if somebody's interested. Yes?
So I have a question about when you were talking about creating the construct itself as well as for the manufacturing. If you're manufacturing more for preclinical uses, and you're identifying candidates, like you mentioned, a lot of the are you able to find them in the lab? Do you outsource the same kind of candidates, or adequate outsourcing?
We do a lot in lab, but there are companies that, particularly for like the NGS work, that you can outsource to. And I know the company's old name, but it's changed recently. It's called Gene Work, but they were acquired by a US company, and they basically hold this field of the NGS testing for herself, DNA, and other impurities. There's definitely CROs that can.
Another question? I have another question about the one dose versus the multiple doses. So what you can comment about that? So it's clear that we can have AAV therapy in one dose, or we require several doses.
I think at this stage it's clear that we should be aiming for one dose, because once you see the immune response after that and the massive spike in antibodies, it will potentially be very hard to remove those and be able to do it through again. We are looking at trying to identify capsid variants that could escape those antibodies, but the load of those after AAV gene therapy is so high, it will be very challenging. There's technologies around trying to remove antibodies such as IDS, but these are sort of early stages, but it is a place that people are trying to look at that sort of dosing and redosing in space.
Any other question? Thank you very much for everybody to come in. Thank you, Dr. Dane, for your presentation.